Okay, so good morning, everyone. I'm delighted to be presenting some of our work here at this wonderful meeting. My name is Thomas Südhof. I work at the Stanford University in California, and I'm an investigator of the Howard Hughes Medical Institute. What I'm going to talk to you about today is our approach of trying to understand human disease, our way of analyzing human neurons. And in order to tell you a little bit more about it, let me tell you that this is part of a broader effort in my laboratory to uncover the biological consequences of genetic changes that predispose to neuropsychiatric and neurodegenerative disorders. I want to illustrate in today's talk how we do this by describing to you two examples that show what we can do with this approach and why it is important for progress in understanding, especially neurodevelopmental disorders, such as schizophrenia or autism. And I hope that with this very scientific approach, you will glimpse a bit of how we, as neuroscientists, can help understanding of what happens to these patients in these devastating disorders. So what I'm going to tell you about is mentioned about two projects. The first project deals with neurexin mutations, the second with synapsin 1 mutations. And in each case, I will explain to you what these actually are. So what are neurexins and why one particular gene, neurexin 1? Neurexins are presynaptic adhesion molecules. In the brain, all communication between neurons happens at synapses, and that communication processes the information that enters into the brain, and that communication in the end produces the actions that leave the brain. Neurexins come in two principal isoforms that I'm not going to discuss in detail, alpha and beta neurexins that differ in their structure but are transcribed from the same genes. There are three genes, each of which has independent promoters for alpha and beta neurexins. Now I'm going to talk to you about neurexin 1 because there's thousands of patients with neurexin 1 mutations, and these patients are usually subject to neurodevelopmental disorders, to different disorders, to autism, Tourette, or schizophrenia, in addition to others less frequently. What is interesting here is that in recent years, there has been enormous progress in the genetic understanding, the genetic landscape of these neurodevelopmental disorders. And let me illustrate this to you by showing you this slide here, which depicts the genetic landscape of schizophrenia and was taken out of a paper that is cited at the bottom. And what you see here is a plot of the odds ratio, that is the probability of getting schizophrenia versus the frequency in the population on the x-axis. And you can see that there's many changes in the genome shown on the very right that have tiny effects. They basically don't cause much of a predisposition. They rarely cause disease. And then on the left, you can see mutations that are rare, but have huge effects, big effect sizes. And among these, in particular, are neurexins, but also many other mutations as well, illustrating that schizophrenia, as you probably can realize, is not a simple disease, but is a disease that is caused, can be predisposed to by many different genetic changes. The problem here is that for most of the affected genes, we don't know the functional impact of a heterozygous genetic variation. And even for well-known genes, the actual functions are often unclear. And this also applies to neurexins. The neurexin 1 mutation is shown here in circle now, demonstrating that it is relatively frequent, although not still rare, and that it has a big impact, but not as big as some of the other mutations. And the neurexin 1 mutation is characterized by deletions. These are heterozygous deletions that delete the neurexin 1 gene to different extents. and that are associated, as I already mentioned, with a variety of neuropsychiatric disorders, especially autism, schizophrenia, and Tourette syndrome. So these different mutations are associated with different diseases. And as it turns out, in these thousands of cases that have been described, there's no clear correlation between the exact mutation and the disease. In other words, any mutation can predispose to any disease, and we don't understand why that is. So the big question is, do these heterozygous deletions produce a meaningful, reproducible biological effect that is independent of genetic background? Are they relevant, or is this an epiphenomenon, especially since they're heterozygous, so there's another normal allele in the patient present? And so to test this question, we used human neurons that we generate from ES cells. And we first generated conditional mutations in ES cells, mutations in which there is an ES cell that has a normal two neurexin-1 genes, but one gene is conditional mutant. And if we add Cre recombinase, we turn this into a real mutation. And using this, we can create neurons that are perfectly matched. But one of them lacks one of the alleles of neurexin-1, the other one is perfectly normal. And what you see here, that these neurons look phenotypically normal. They have a normal morphology, they form synapses, but they express, as you would expect, as shown at the bottom, less neurexin-1 mRNA. They are truly mutant neurons. And so these neurons afford us with the opportunity to study how the neurexin-1 mutation affects neuronal function, if it does at all, because after all, it's heterozygous. We found, as mentioned before, that there's normal development of the neurons themselves. There's normal synapse numbers. So we tested for synaptic transmission, synaptic function. And what we found is shown in this slide here, namely that when we looked at evoked synaptic transmission, there was a significant decrease in synaptic strength that corresponded exactly to the decrease in neurexin-1 mRNA levels. When we did repeated stimuli, as shown here on the right, we found that this selective decrease in synaptic strength only applied to the first response of a stimulus train, suggesting that subsequent responses normalize. And this discrete but robust phenotype was quite surprising because it demonstrated that there was a selective effect of this mutation on synaptic transmission, on how synapses process information, and suggests a possible defect, a possible impairment that might be amenable to pharmacological intervention, and thus is important. So the next question we asked in this project then was whether the same change could be observed in patient-derived neurons, i.e., whether this is just a phenomenon that we instituted by using a artificial mutation, or whether patient-derived neurons have the same alteration, impairment in synaptic transmission. And we were particularly interested in using a rigorous approach to test this because there is, as you probably know, a lot of problems about reproducibility in science these days. And so we were interested in doing this in a manner that controlled for every possible variable. And so to do this, we performed the following design. We followed the following design. We used a cross-lab, cross-platform validation and analyzed human patient-derived neurexin-1 deleted neurons. We obtained pairs of ES and IPS cells from patients, from human patients and control subjects. These were frozen and the IPS cells were then distributed to different centers, namely to one at Stanford and another one at the FCDI. They were then transformed into neurons and they were analyzed functionally by different labs in different institutions at Stanford and at Rutgers. And using this approach, we found again that neuronal development was normal. But strikingly, when we tested synaptic function, we observed the same impairment that we had seen previously with the engineered mutation. And this is summarized in this one slide here. I'm not going to go through the details because that is maybe a bit too granular for this audience. Let me just focus on this one middle section here to illustrate to you that in this analysis that were performed in Stanford and the same results were obtained at Rutgers University, we find on top, again, the same decrease in synaptic strength that I illustrated to you earlier, with about a 40% decrease in the amplitude of evoked synaptic responses. This is associated with an increase in the coefficient of variation, which indicates a decrease in release probability. And what you see at the bottom is the so-called paired pulse response curve that illustrates that there is a decrease in paired pulse depression, which again illustrates a decrease in release probability. So what we see here then is that there is a massive impairment in synaptic transmission that is induced by this heterozygous mutation, and that it might explain the predisposition of these patients to neuropsychiatric disorders. In summary, what this particular project illustrates is that heterozygous human neuroexin-1 mutant neurons exhibit a robust synaptic impairment that could serve as the basis for mechanistic and translational studies. This doesn't explain why different patients have distinct phenotypes, which I believe is actually a very important question that might depend on additional mutations in the genome in different patients or genetic variation. However, it does elevate the neuroexin-1 mutation into a validated pathophysiological mechanism that changes how synapses process information and thereby predisposes to neuropsychiatric diseases. Let me move on and tell you about a completely different second example, namely Project 2, analysis of synapsin-1 function. This is completely different because the biology is different here. Synapsin-1 is a synaptic vesicle protein. In fact, it's the first synaptic vesicle protein that was identified in the laboratory of the late Paul Greengard in beautiful work dating back 40 years ago. And we cloned this protein initially as one of our synaptic vesicle cloning projects 30 years ago. The function of synapsin-1, however, to this date remains controversial. It is interesting from the viewpoint of human disease because what happens is that in some patients, mutations in synapsin-1 have been observed that predispose again to autism and intellectual disability. And these mutations are shown here in the context of the synapsin-1 gene. What you see here is the domain structure of synapsin-1 shown in different colors and the mutations on top of it. Synapsin-1 is a large protein that has multiple domains. At the N-terminus, there is an A domain that is shown here in red that is followed by a B domain, a C and D, and an EF domains. What's important is that the C domain is an ATP binding domain that we demonstrated earlier has an ATP binding cassette, as shown by crystallography. Whereas the A domain, importantly, contains a conserved phosphorylation site that is the substrate to protein kinase A, and that is regulated by pKa-dependent phosphorylation. And this will be important in my subsequent analyses that I will show you. So to analyze what the effects of these mutations might be on human neurons, on synaptic information processing, and in general, what the function of synapsin-1 might be, we again generated conditional mutations and generated conditional knockout neurons. What you can see here in this slide is a validation of these conditional mutant neurons. On the left, you can see an immunoblot that illustrates that after addition of Cre, there's a loss of synapsin-1, demonstrating that these conditional mutant neurons indeed delete synapsin-1 expression. What you see on the right is that when we mutate, when we generate these neurons develop fine, even in the absence of synapsin 1, shown on the right. But you can easily see again that synapsin 1 is absent on the right because the green synapsin 1 on the left is completely gone on the right. So again, we created a tool whereby we can analyze in neurons that are genetically identical except for the synapsin 1 mutation, the effect of the deletion of synapsin 1 by comparing identical neurons either with or without synapsin 1. And so again, there was no significant effect on the morphology or development of these neurons, and so we focused on function. We focused on synaptic transmission because after all, synapsin 1 is a synaptic vesicle protein. And we analyzed this again by electrophysiology. We tested this in conjunction with forskolin because forskolin activates cyclic AMP synthesis, which then in turn activates pKa, which then in turn induces phosphorylation of synapsin 1, as I described to you earlier. And what we found is shown in this slide here. What you see here on the left are sample traces in which we depict synaptic responses in these human neurons with or without synapsin 1, with or without forskolin. And what you can see here on the right is in a typical example that when we just delete synapsin 1 without forskolin, the blue trace demonstrates that there's a decrease in the amount of neurotransmitter release during this tremulous strain. So this is the exact opposite of what we see with neurexin 1 mutations. In neurexin 1 mutations, we see that the first response is abnormal, but then it normalizes. Here, we see that the first response is normal, but then it becomes abnormal. It responds much less strongly. So the opposite is complete. The phenotype functionally is completely different, but it's just as robust as you can see at the bottom in the middle, where you can see the cumulative response plotted in nanoamperes, so absolute responses, where there's this massive decrease as the stimulus train proceeds. Now, strikingly, when we add forskolin, the difference goes away. And it goes away not because there's a change in the synapsin 1 deleted neurons, but because there's a change in the wild-type neurons that is not present in the synapsin 1 deleted neurons. In other words, in the presence of forskolin that activates synapsin 1 phosphorylation, the difference between synapsin 1 mutant and wild-type neurons goes away because the wild-type neurons become like a synapsin 1 mutation. It's as though forskolin induced a synapsin 1 deletion by itself, which obviously it doesn't. So synapsin 1 deletion and forskolin treatment induce synaptic depression, and the synapsin 1 deletion occludes, prevents the forskolin effect, suggesting that forskolin acts by inhibiting synapsin 1. How does forskolin phenocopy the synapsin 1 deletion? What happens? And now I want you to recall that synapsins are cyclic AMP dependent protein kinase substrates, which I showed you earlier, where there is this phosphorylation site at the end terminus of synapsin 1. So pKa-mediated phosphorylation controls binding, as it turns out, of synapsin 1 to synaptic vesicles. And this is something that we actually showed 20 years ago when we didn't really know what synapsin 1 does. But we demonstrated, as shown here in this old slide taken from a paper in 1999, that N-terminal phosphorylation of synapsin 1 controls its binding to synaptic vesicles. What you see here on the left is the phosphorylation of synapsin 1, and what you see here on the right is its association with synaptic vesicles. And what you can see is that when you add cyclic MP, as shown here, phosphorylation goes way up, as demonstrated with this dark bar. And when you add an inhibitor of pKa, obviously phosphorylation is blocked. When you look at the right in this barred, in this framed part of the slide, you see that cyclic AMP increases the dissociation as it increases the phosphorylation. So synapsin 1 dissociates from the synaptic vesicles, and this can be prevented by blocking cyclic AMP dependent protein kinase A. So this suggests a pathway whereby forskolin induces the dissociation of synapsin 1 from synaptic vesicles by phosphorylating synapsin 1. And it suggests that it's the synapsin 1 that's on the vesicles that's active. But when you dissociate it, it becomes like a deletion. So what does this actually do? How does this work? What is the reason for this change in synaptic transmission? So to test this, we used electron microscopy, and we used a special kind of electron microscopy that preserves the structure of human neurons better, fast freeze electron microscopy, in collaboration with Christian Rosenmund's lab in Germany. And what you see here are images of such fast freeze electron microscopic pictures, demonstrating that you can see beautiful synapses which contain tons of synaptic vesicles in the presence of synapsin, as shown in the middle panel, but seem to contain less vesicles in the absence of synapsin, as shown on the right panel. And when you quantify this, you see that in the absence of forskolin, as shown here in this framed bars on the left, the synapsin 1 deletion decreases the number of vesicles per terminal dramatically. So it makes the vesicles disappear, which explains why there is an increased depression. When you add forskolin, as shown here in the right panels, forskolin alone decreases the vesicles. It basically makes the vesicles disappear. And then, and the addition of synapsin 1 deletions has a very small effect, consistent with the fact that the synapsin 1 deletion occludes the effect of forskolin. So what happens here is that the synapsin 1 deletion or forskolin makes vesicles go away from terminals. Now, these vesicles are not destroyed. Instead, they are more distributed diffusely in the nerve terminals so that they no longer form the cluster right at the release sites. And they're not available for release anymore. In other words, the reserve pool of the vesicles for release becomes diminished. And as a result, there is more depression. So this illustrates, reveals the function of synapsin 1 and also demonstrates what happens in the absence of synapsin 1. The synapsin 1 deletion and the forskolin-independent phosphorylation of synapsin 1 decrease vesicle numbers, and thereby synapsin 1 deletion occludes forskolin. There's no general loss of synapses. The synapse number remains the same. It's the synaptic vesicle loss. And we can visualize the synaptic vesicle loss directly, even without electron microscopy, by immunofluorescence staining intensity. This is illustrated here, where you can see on the top immunolabeling of mutant and wild-type human neurons or two synaptic vesicle markers, synaptotagmin 1 and synaptophysin, both of which we cloned decades ago. And what you can see here is that either forskolin or the Cre-dependent deletion of synapsin 1 decrease the signal for these two synaptic vesicle proteins in the nerve terminal, as you would expect when the vesicles go away. What you can see in the bottom, however, is that a non-synaptic vesicle marker, HOMA1, remains just normal, demonstrating that indeed the synapses themselves don't change. What changes is the synaptic vesicle numbers. And so this reveals a regulatory pathway that controls synaptic vesicle numbers in presynaptic terminals. Forskolin induces pKa-dependent phosphorylation of synapsin 1, which then causes synapsin 1 dissociation from vesicles, which in turn produces dispersion of synaptic vesicles. Now, this is all very nice, I hope you believe it, but it's very artificial. Forskolin is not something that normally happens in the body. In fact, it's highly toxic. You would never want to give that to a person. So the question then arises whether this discovery of a pathway is physiologically relevant. Does this ever happen in a real synapse in a brain of a person. And to address this, we were reminded by our colleagues that there are presynaptic neuromodulators, which are many of them, such as norepinephrine, serotonin, dopamine, many others, that regulate the state of the mind and that act on presynaptic terminals to regulate cyclic MP levels in the presynaptic terminal. And so this raises the question whether these presynaptic modulators could control synaptic transmission by a synapsin phosphorylation-dependent mechanism. And so in order to address this question, we first looked at the phosphorylation of synapsin 1 in human neurons as a function of these neuromodulators. And what we found is that serotonin induced a strong phosphorylation of synapsin 1, whereas, for example, norepinephrine, even at very low levels, 1 micromolar, caused a suppression of the steady state level of phosphorylation of synapsin 1, demonstrating that these neuromodulators indeed can modulate the phosphorylation state of synapsin 1 in a living neuron. And we can mimic this using drugs that are alpha-2 receptor agonists, as shown here on the right, and that change phosphorylation of synapsin 1 by stimulating the norepinephrine receptor. So we can use these drugs to actually manipulate the nerve terminal phosphorylation of synapsin 1 as well. And so we then asked whether does neuromodulator control of synapsin phosphorylation control vesicle numbers? And is this control of vesicle numbers dependent on synapsin 1? What you see here is my final data slide that illustrates to you that indeed, this is exactly what happens. This is a physiological process. When we look at the synaptic vesicle numbers as measured by synaptophysin or synaptotagmin puncture density, you can see that the agonists of the norepinephrine receptor cause an increase in synaptic vesicle numbers. And you can also see that upon deletion of synapsin 1, as shown in the orange bars, this is gone. There are just fewer vesicle numbers, and the norepinephrine agonists no longer have this effect, as shown by 2 different markers. When you do the same experiment by staining the neurons for synapsin, as shown here in the middle, you can see that in the presence of synapsin, again, there's an increase in synaptic vesicles, as now shown with synapsin immunoreactivity. But because we're deleting synapsin, as shown on the right, there's only a background signal after deleting synapsin. And finally, as a control, as shown here on the right, when you look at synapse numbers, as shown with a general non-synaptic vesicle synapse marker, in this case, Bassoon, there's no change. So synapse numbers don't change with the presynaptic neuromodulators. It's specifically the synaptic vesicle numbers. And further experiments demonstrated that this applies to multiple neuromodulators, most importantly, possibly to neuromodulators that are based on endocannabinoids, which are very important, maybe the most important neuromodulators in brain, and that also operate in part by this novel mechanism. So what I want to do now is summarize to you what I've tried to tell you in this part of my talk. What I've tried to tell you is that we observed a novel pathway of regulation whereby presynaptic neuromodulator receptors activation causes either an increase in cyclic MP, such as, for example, produced by serotonin. This induces an increase in synapsin phosphorylation. Synapsin dissociates from synaptic vesicles disperse from the cluster, and there's decreased neurotransmitter release during sustained stimulation. On the other hand, if we decrease cyclic MP, as observed, for example, with norepinephrine or with endocannabinoids, marijuana, what we see a decrease in synapsin phosphorylation. The synapsin associates with synaptic vesicles, synaptic vesicles move into the vesicle cluster, there's more synaptic vesicles, and there's an increase in neurotransmitter release during sustained stimulation. And this mechanism of regulation is absent in patients where there is a mutation in synapsin 1 that causes autism and intellectual disability, suggesting that this is a universal mechanism that is important for understanding neuropsychiatric disorders. I've reached the end of my presentation. What I try to tell you about today is about two different projects that are very different. They're united by analyzing in human neurons, not mouse neurons, not rat neurons, human neurons, mutations that are pathophysiologically relevant and that are predisposing patients to neuropsychiatric disorders. One mutation, the orexin-1 mutation, changes synaptic transmission by decreasing the release probably initially during a train of action potentials. The other mutation, the synapsin-1 mutation, changes synaptic transmission by decreasing the release probability, not initially, but later in the train of a series of action potentials. They have completely different mechanisms, but in the end, in terms of the patients, they have similar deleterious consequences. And I would like to close this scientific presentation by acknowledging the people who performed this work, most prominently Chang-Hee Pak for the orexin-1 mutation in collaboration with Tamas Danko, and Christopher Patzke for the synapsin mutation. And this was what carried out with my colleague at Stanford, Marius Wernick, at his laboratory, as well as outside collaborators who I don't have time to name. Thank you very much for your attention. I hope this has been informative and interesting. Thank you.
Мы – транспортно-логистический холдинг «Российские железные дороги». Мы развиваем транспортную инфраструктуру страны, строим новые станции, вокзалы и создаем новые возможности для вашего бизнеса. Лучшие скоростные и высокоскоростные поезда сделают ваше путешествие комфортным и безопасным. Мы – локомотив экономики. Сотни миллионов тонн грузов ежегодно. Развитие науки и социальной сферы – это наш вклад в благосостояние страны и ваша уверенность в завтрашнем дне.
Я новая Hyundai Creta. Hyundai.
Есть история фантастическая. Есть история сказочная. Есть страшная история. А есть история реальная. В Юка всегда реальные истории и диагностика автомобилей с пробегом. Юка – официальный сервис Hyundai. Смотрите историю и диагностику автомобиля и покупайте его онлайн.
If you want to stay ahead of the competition, you need the most advanced solutions to transform your comms performance. From media database to social influencers, distribution to monitoring, analytics to insights, Cision's cloud-based tools can help you communicate like never before.
Звонки ВКонтакте для работы. Больше никаких ограничений по времени и платных функций. Звонки ВКонтакте – это разговор